Welcome back, everyone. Today we are looking at the cell part 2. This is still in chapter 3. And this is your reading sections and then the review questions if you want to look at those. And hopefully you've already read over the key concepts and key terminology. And our question of the day is, how fast are sperm cells? Ooh, intriguing. How fast can these little guys go? And we'll find out. So for the pre-lecture assignment, I am going to go through it. I usually don't, but it's important that you guys know all of these cell parts or organelles. So if you already feel comfortable with the functions of these, you can skip this part, but I will go ahead and go over it with you. So the nucleus, the function of the nucleus is to store genetic information, as long chains of DNA. Store genetic information as long chains of DNA. So in our picture here, the nucleus is located right here. And in high school or middle school, possibly, you probably heard of the nucleus as being the control center of the cell. And that's because it has the DNA and it has all the instructions for making proteins, which are required to carry out all the processes of the cell. The nucleolus is located within the nucleus, so in this picture it's this darker spot here. And this is where ribosomes are made, which are very important organelles, and we'll talk about those in just a minute. The nuclear envelope separates the nucleus from the cytoplasm. So the cytoplasm is this jelly-like substance that kind of holds all the organelles in place and fills the cell. And you can see there are these nuclear pores in that envelope that allow things to come in and out, because obviously we need communication with the control center of the cell, which is the nucleus. And it is continuous with something called the endoplasmic reticulum. And sometimes you'll just hear this referred to as the ER. And that's the structure that you can see here. So ribosomes and proteins move into and out of the nucleus right here through those nuclear pores. Okay, so what are ribosomes? They are composed of proteins and rRNA. So ribosomal ribonucleic acid, one of the nucleic acids that we talked about in our macromolecules chapter, and its job is to synthesize proteins. So it makes proteins, so they're kind of a big deal, they're important, and they are floating around the nucleus or attached to the endoplasmic reticulum, which we're getting to in just a second. So there are ribosomes just kind of around in the cytoplasm, and then they're also attached to what we call the rough ER here, the rough endoplasmic reticulum. So the endomembrane system You have the rough endoplasmic reticulum, which is studded with those ribosomes. And I don't know why this makes me think of the word bedazzled, if you're familiar with that, but just like has all the ribosomes just stuck within it. And proteins enter the rough endoplasmic reticulum and are packaged into vesicles, which are just like little sacs for transporting materials and molecules. So with the endoplasmic reticulum, there's rough and smooth. So the rough is studded with ribosomes, proteins are packaged there, and the smooth synthesizes or makes phospholipids, and other lipids. So I don't know if this will help you remember, but I think smooth like butter. I don't know if that's in a song or what or why I even have that in my head. But the smoothie makes phospholipids and other lipids, basically fats, which is butter. So smooth like butter. I don't know if that will help, but that's how I remember it. And another part of the system is the Golgi apparatus. So that is involved in the processing, packaging, and secretion of proteins. And I'm going to circle the word secretion because that's going to be important later. And another organelle are lysosomes, and they contain hydrolytic enzymes. So if you haven't learned this yet, an enzyme is any protein that speeds up or facilitates a chemical reaction, and they contain these hydrolytic enzymes. And if you remember back to hydrolysis, that's when you break molecules apart, and so they break down foreign matter. And they also perform something called autodigestion, where parts of the cell are broken down. So I don't know if this is related in any way, but again, it's how I remember it is lysosomes. That's kind of like Lysol, the cleaning product. And so they're kind of like cleaning up the cell. They break down foreign material that shouldn't be there. They start to digest it and break it apart and essentially get rid of it. So lysosomes, Lysol, cleaning, breaking down. Hopefully that helps you remember. All right, the cytoskeleton. And all my papers are out of order. That's awesome. Sorry, 1 second. Okay, the cytoskeleton is a protein scaffold that provides structure in the cell and helps it maintain its shape. And it locks organelles into stable position. But keep in mind that this cytoskeleton is constantly being moved and remodeled. So again, it's not static. It's not just staying how it is forever. It's constantly being moved around and remodeled for the function that it needs to do. Okay, and we have 3 components of the cytoskeleton, and these will be important to know. So I'm putting a circle there. We've got microtubules, actin, and something called intermediate filaments. So microtubules help to maintain the shape of the cell. And they provide a highway system for vesicles. So I picture it like when you go to the bank and you grab the little canister and you put whatever you're sending to the bank in it and stick it into that tube and push the button and away it goes, up and over, down and into the bank. So think of the microtubules as like the tube that goes through. It's like a highway system for things to get transferred through the cell. And something that a lot of students forget is this is what helps to separate chromosomes during cell division, during mitosis, if you're familiar with that. So it kind of latches onto the chromosomes and helps to pull them apart as the cell is dividing. And they originate from the centromere. Which is just another organelle. It's usually near the nucleus, and they kind of branch out from there and go do what they gotta do. Alright, number 2 is actin, and these line the periphery of the cytoplasm. So remember, the cytoplasm is that jelly-like material in the cell, so it kind of forms a line around it. And it helps with something called cell walking, which I think is awesome that these cells can actually move around using these actin filaments. And this is important in the immune system and white blood cells, so they're able to move around and gobble up whatever it is that they need to eat. Sorry, I'll try to not talk so much with my hands. But yeah, that is actin. And then the way that they work is they slide relative to each other. To achieve movement. And when you get to physiology, if you ever take it, you'll become very familiar with actin and myosin, which are involved in muscle contraction. And the third and final part of the cytoskeleton is called intermediate filaments. These provide structure for the nucleus. and they tether or hold cells together. So I don't have the color copy, you guys probably do, but here we have 1, 2, 3, 4 cells, and the green is showing the microtubules, which again help maintain the shape of the cell. They provide the highway system, help separate the chromosomes during cell division, and the actin is white, which is going to be lining the periphery of the cytoplasm and help with cell walking. And then the nucleus is the blue portion, which is where you'll see a lot of the intermediate filaments. Because they are providing structure for that nucleus. Okay, so cytoskeleton, constantly being moved and remodeled, protein scaffold, structure for the cell, microtubules, actin, and intermediate filaments. And we'll come back to these three in just a minute. Or right now, actually. So in class problem set. So this says several human disorders are caused by abnormalities in cytoskeletal proteins. In addition, cytoskeletal proteins are targets for many drugs. Read each description from medical literature and decide which skeletal protein out of the three that we learned, microtubules, actin, or the intermediate filaments, is the cause of the disorder or target of a specific drug. Now, this was not written by me. This is really hard, and it's not going to be this difficult on the exam. So when you look at these, don't panic. If you want to pause and read through them on your own right now and try to decide what goes where, microtubule, actin, or intermediate filament. But I'm just going to go ahead and read through it and give you the answers. So, epidermolysis bullosa, sometimes called EB, is a group of rare inherited skin disorders that are characterized by the development of blisters following minimal pressure to the skin. Blistering often appears in infancy in response to simply being held or handled. In rarer forms of the disorder, EB can be life-threatening and there is no cure for the disorder, so the treatment focuses on preventing and treating the wounds and infection. And I did a little research on this. This is a really, what's the word I'm looking for? A really devastating disorder. So what happens is basically the skin is very fragile and all it takes is the slightest rub to cause these wounds and blisters. And if you look up pictures of it, it looks like burns. Like I don't even know how to describe it. It looks awful. But this is because the skin, the epidermis and the dermis can't really tether itself together. It can't really hold itself together. And so this is what are we trying to answer here? Which skeletal protein is the cause of the disorder? So this is because the intermediate fibers aren't able to properly do that, or intermediate filaments, I'm sorry. And it's caused by a mutation, so those aren't functioning properly and can't hold the skin together. Down to the other page. Count the layers, that should say, of the skin together. Okay, the next one. Metastatic cancer cells use cytoskeletal bundles to underpin protrusions that allow them to break away from a primary tissue and invade through the surrounding tissue. After traveling in the vascular structure or lymphatic system, they exit into a new niche and seed a new tumor, often after lying dormant for months or years. During metastases, cells adapt their motility and adhesive capacity to suit their environment in much the same way as embryonic cells do during morphogenesis. So I underlined some keywords here. They all have to do with moving around. So if you remember, actin allows for that cell walking we talked about. So these actin polymers allow for cell walking. And we can probably deduce what this last one is going to be, but let's look at it anyway. Paclitaxel, hoping I'm saying that right, sold under the brand name Taxol, is one of several cytoskeletal drugs, and paclitaxel-treated cells have defects in mitotic spindle assembly, so of cell division is what we're talking about here, chromosome segregation, and cell division. Paclitaxel stabilizes the protein polymers and protects it from disassembly. Chromosomes are thus unable to achieve a metaphase spindle configuration, and this blocks the progression of mitosis, and prolonged activation of the mitotic checkpoint triggers apoptosis or reversion of the G0 phase of the cell cycle without cell division. That was a lot of medical terminology. Basically, I think what this is saying is that there's a medicine you can take that this is making it sound like it's trying to fix the problem with the cell division, but everything that I researched about this drug shows that it's used for cancer treatment, because for the most part, or I should say in general, cancer is just uncontrolled cell division where the cells keep dividing when they're not supposed to, and that forms a tumor. And so this is used a lot in chemotherapy to actually go prevent the microtubules from doing their job of helping the cell to divide. How this is targeted and exactly physiologically how it works, I don't know. But again, this is not, you will not be asked questions of this detail. But it is interesting to learn about. Okay, so some cytoskeletal structures. We've got cilia and flagella. So cilia are microtubule projections of the cell membrane, and here we have them, a microscopic image of it, and they're kind of like little finger-like projections, kind of like eyelashes. In fact, I think cilia is Latin for eyelashes. So just picture these sweeping, beautiful eyelashes. And the singular term for cilia is, I think this was written on here wrong. I think it's cilium. And I think I spelled that wrong. I need to look that up. Alright, so again, microtubule projections of the cell membrane. So we've got the cell membrane that kind of folds out like that in those finger-like projections, and they do this kind of oar-like movement. So they're in there like synchronously swishing back and forth, and these have important functions in the human body. They line the trachea, the tube that leads from your throat to your bronchi and your lungs. And it sweeps debris upward. So your lungs are very fragile. You don't want a lot of foreign material and little microscopic pathogens getting in there. And so these trachea the cilia and the trachea kind of swish it out. And then guess what? You cough and out it goes, out of the body. Next, we have flagella. So these are when microtubules form a whip-like tail. And I know what we all think of when we hear flagella, at least me. It's the structure in sperm. And we do see flagella in several other organisms. I don't know how many, but in the human body, the sperm cells are the only place where you will find flagella. So, sorry ladies, you don't have any flagella in your body. So to our question of the day, how fast do these guys move? Well, they are actually quite impressive. So, sperm can move 5 millimeters per minute. You might be thinking, wow, super fast, 5 millimeters per minute, way to go, guy. But it's actually 5 of their body lengths per second. Now, if you really want to be impressed, and to put this into perspective, if this sperm were blown up to the size of a salmon, it would be moving at 500 miles per hour. And it pains me to say miles per hour because in my 7th grade science class, like miles and inches and feet and pounds are all swear words. So I should probably convert that to kilometers one day, but I think 500 miles per hour is probably more relatable, so I'll allow it. But they are fast. And they need to be because they're all racing to get to the egg first. There'll only be one winner, and it's going to be the fastest and the strongest. And here's another in class, I'm going to call it an online individual problem set. So with this one, you're going to want to refer back to your PreLecture that goes over the functions of all of the organelles. Let me find that in our pack here. Right here. This is going to help you out, so I'm going to set it off to the side here. Alright. So again, if you want to pause this and do it on your own, now's the time. If not, follow along with me. Cell structures discussed are found in every cell, but some cells will have a greater or lesser abundance of some structures depending on the feature. Working in groups, determine what specific cell structures will be overrepresented in the following specialized cells. So first up we have Leydig cells. These are found in the testes and they make and secrete testosterone. So we have a keyword here, secrete, and testosterone. So thinking back to our macromolecules, a testosterone is a steroid, so it falls under our, I believe our triglyceride, no, what are the fatty acids? Lipids. Falls under the lipids category. So, the overrepresented organelle, what are these Leydig cells going to need a lot of in order to do this function? Well, let's look back at our sheet here. The organelles that secrete are the Golgi apparatus, so these Leydig cells, a lot of Golgi apparati. Is that the plural? I have no idea. Secrete. When I try to talk and write at the same time, I mess up, and I'm sorry. And lipids, smooth like butter, they're going to have a lot of smooth endoplasmic reticulum. That makes that testosterone. That is a lipid. And these are more like the type of questions you'll be asked on the test. Similar to this. The exam. Sorry. I confused my middle school educational jargon with the university level stuff. Salivary gland cells. So these make and secrete enzymes into the mouth that break down starch. So secrete, when you hear secrete, what are you automatically going to associate that with? The Golgi apparatus. Which processes, packages, and secretes proteins. And then enzymes are proteins. And so the rough ER, which is studied with those ribosomes that make proteins, going to need a lot of those. Ribosomes, I'm sorry, my handwriting, guys, that make proteins, which are enzymes. Alright, next up, white blood cells. They destroy foreign material within the body. So, one keyword is destroy. So remember the cleaner uppers of the cell, the lysols of the cell, are the lysosomes. They're going to break down foreign material, which is pretty important in immunity. And this might not be outrightly obvious, and my picture isn't very good, but if you look, they've got all these little, I'm going to call them feet, to help them walk around to where they need to go. And remember, this is actin that helps with cell walking. So in our white blood cells, we have an excess of lysosomes to help break things down, and a lot of actin because those cells have to be able to move around the body to help get rid of pathogens and foreign material. Muscle fibers. These are for movement. They contract, so movement, got the actin. They kind of, there's these structures in there, I think it's forget the names right now, but they kind of slide past one another in order to allow those muscles to contract. You have a lot of protein in your muscles, so we'll need a lot of ribosomes to make those proteins. And we haven't talked about this organelle yet, but something called the mitochondria, and I'm sure we're all familiar with the saying. The mitochondria is the powerhouse of the cell because that's what makes energy, and your muscles use a lot of energy, and if you look at muscle cells underneath a microscope, you can see they have an abundance of mitochondria. And like I said, we haven't talked about this yet. That's going to be in our next lecture on cellular respiration, which is quite one of my favorites. So we'll get to the mitochondria. And is that the end? That went quick. That was a fast one. I'm sure you guys aren't too upset about that, but next time, the last lecture of the first module will be next on cellular respiration, and that one is quite intense and will probably take longer, so we won't feel bad about making this one short because the next one will be a little longer. As always, please, if you have questions, I'm here to help. That's my job. It's hard to do online. I'm not used to it. I'm used to being there in person to immediately help. So please, if you are confused on something, have any questions, message me on Canvas, send me an email, and we'll set up something to help you figure it out. All right. Have a good day.